Hello, my name is Ankit Singh and I am going to show you my approach and analysis, how I perform the analysis of gene and phenotype embedding. So first, here I imported all the data and then phenotype gene embedding. First, I loaded the phenotype gene data for open target step 2 for LLM PhD. And after loading it, then I loaded ground truth data, and after ground truth, I concatenated these two data. So after concatenating it, both of them have same number of rows. So rows information in each row were aligned with each data frame. So I concatenated them, and after concatenating the data, I form a hashing function. So in hashing, I use my name to get hash value, and after generating hash from my name, I took the modulus of 2 to the power 32 because the hash generated by my name was too much large for Python to take it as C. Then on the basis of hash value, I generated my unique data set, and after generating my unique data set, I applied. So after generating my unique data set, I splitted the roles. So symbol gene string has so many genes for one single phenotype. So I for each phenotype I split the rows. So after splitting the rows, so I got like AR3D, E1, FD. So all the genes get split, and the genes which are common in symbol and symbol gene list, because symbol is from this column is from ground truth data. So here if these two columns are common, these two rows have the common gene name, then it is true, means this is a causal gene of the type 2 diabetes, and others above these four genes are not the causal genes. So I use this approach to classify the data as causal and non-causal on the basis of genes. So here perform the data as causal and non-causal. So here we can see this gene and this gene are same in the same row, so it is causal, and other genes are not same, so these are non-causal. Then I drop the few columns which are of no use, and check like if we have any value, so there are no missing values. So we are good with the data and the save the data. Then I loaded the gene embedding data, and after loading gene embedding data, I merged it with my final data. So for what basis I merged these two data? So I merged this data on the basis of symbol gene list and zero column. So these two columns were common. Just the name of the column is different, but the information stored here is common. So I use these two columns to merge the data. So here I use merge function, pandas merge function to merge the symbol gene list on right, zero out inner. So I merge them, and after merging these two data sets, so I got this data set as embedding for every gene. And then I have label encoded like zero for non-causal, causal, and one for causal gene. So what happens is now I started visualizing the data and perform t-sign and various other dimension relation techniques to find the relationship between causal and non-causal genes. So here first I generated the embeddings, took out embeddings, and make another data frame of the gene embedding. So first I applied t-sign function. So first applying after t-sign function, we see this graph. So this figure shows dense distribution of points across the t-sign space, indicating a wide variety of gene embedding. And here we can see like causal genes are represented by orange color and non-causal genes are represented by blue color. And we can see in this graph that there is a significant overlap between the two groups, like with many points from both categories appearing in similar regions. The space suggests that these embeddings are similar uh, because they are lying in the like they are overlapping with each other, indicating that these features are used to generate these embeddings do not distinctly separate the categories. So here we can easily see that these two features are not separating the categories uh, very well. So but we can see like there are also non-causal genes which are forming different clusters. So these gene embeddings are forming different clusters. So we can say like these have different embeddings or different properties. So they are forming different gene clusters, but most of the causal genes and non-causal genes are overlapping. So we can say like these embeddings of the causal genes and non-causal genes are not so much different from each other. Like they don't have any not much variation. And one possibility can also like be that causal and non-causal genes are acting together. Means these are cascade of actions, like causal genes and non-causal genes are acting together for one phenotype. So here are the observations and inferences that I have derived from the data after seeing the plot. So you can clearly see the close proximity of causal and non-causal genes in T-sign space suggests that these embeddings are similar. Yeah, so we talked about that. And then we come on UMAP projection. So here in UMAP, the genes, here we can also see the causal and non-causal genes are overlapping with each other. There is not much difference between each other. And this is UMAP embedding projection of gene embedding with their names. So it is pretty much matched up. And then in PCA, after applying PCA, we can see like a few of the genes are forming non-causal genes are forming clusters, but here we can see causal genes are still overlapping with non-causal genes. So we can see this is not very much different. And after that, we applied 2D UMAP with k-means clustering, and after applying 2D UMAP with k-means clustering, we can see like here causal genes and non-causal genes are centered, clustered at the center, but the non-causal genes are causal genes. Many of the causal genes are spread across the outer area. So at center we can see many causal genes are clustered together. So further, when we plot graph with phenotype, we will see that these graphs, these embeddings, how these embeddings are performing, acting for a particular phenotype. After that, we perform ability clustering. We can say hierarchical clustering also. So in hierarchical clustering, we can easily see that the causal genes are forming clusters separately from non-causal genes, but still these are surrounded by the non-causal genes. So after applying many clustering techniques, we can easily say that these causal genes and non-causal genes don't have very different embeddings. Their embeddings are almost similar. Up to certain level, their embeddings are almost similar, and they are acting together. Like a particular gene is not responsible for any phenotype, because not a particular gene is forming a phenotype, because, but many genes acting together for the phenotype, because the distribution is distributed. Still here there is cluster, but it is surrounded by the non-causal genes. And after that we plotted PCA. So to see how many components we need, so I took 200 component which has 83.96% variance. And after performing PCA, I performed linear discriminant analysis. So after performing on linear discriminant analysis, we can see like class 0, which is non-causal, has higher density around the values near 0. So it has higher density, while one class 1, which is causal, it has spread out more distribution. So it is spread from -1 to 6, whereas it has higher density towards 0, class 0, or non-causal genes have. So this overlap between the two classes suggests that some degree of similarity in their distributions. As we already said that their embeddings are not so different. They are somewhat similar. So there is a distribution similarity in their distribution, and this indicates the areas where these classes, like genes or classes which are not clearly separable. So and the density of class 0 is higher in the negative region, while class 1 is more concentrated towards the positive region, indicating that it is a potential for good classification models. Then here we applied PCA. So this PCA graph we already saw. Here I tried to use different parameters, but still got no better result. Then after this I used supervised UMAP. So in supervised UMAP we provide the label column also. So after providing label column, we can see that certain class 1 means causal genes are forming clusters, small clusters, whereas non-causal genes are forming dense clusters. So we can clearly say that these causal genes and non-causal genes like forming dense clusters uh, away from each other, but we can see the outer area away from the center, that these genes are not much separated. So you can see like we can again say these genes are not so much different in their embeddings. They share similar space, and these are the conclusions. You can read them or analyze them. Combining gene embedding with phenotype embedding. So up till here we analyze the relationship between the causal and non-causal genes. So from here we are going to analyze the relationship between gene embeddings and phenotype embedding. How a particular embedding is distributing in phenotype embedding. So first I rename them because I have to merge the phenotype embedding data in my final data. So after renaming, I merge the final phenotype data. So phenotype data is merged, and after merging them, I converted the genotype. After merging them, I converted the column which contain the embedding into list form here, and after converting them into list form, I converted them into data frame. So this is data frame gene embed, which shows genotype embedding. This is pheno embed, which shows phenotype embedding. And after this, I plotted a graph for phenotype embedding to see how much components we should take. So I took at least 200 components, and the 200 components were retaining 99.81% of variance, so it was good. So after applying this, I performed UMAP and T-sign, and then plotted graph for phenotype and gene embedding. So after performing graph for phenotype and gene embeddings, we can clearly see that here, like gene embeddings and the phenotype embeddings are overlapping with each other. So this overlap between gene and phenotype embedding suggests that there are certain genes which might be linked to specific phenotypic traits, and this could be a starting point for further investigation into gene phenotype relationships. The distribution of phenotype embeddings indicate greater diversity in phenotype traits, because the phenotype embeddings are scattered everywhere, but we can see like genotype gene embeddings are clustered in the center. This could imply that phenotypes are influenced by a broader range of genetic factors or environmental conditions also, and there is not much similarity between genotype and gene embeddings and phenotype embeddings. Then after that we visualize T-sign embeddings, and these T-sign embeddings also we got the same influence, like many of the phenotype embeddings are being governed by many genes, not by single gene. So here we can see like say that a single gene is not only responsible for phenotype embedding, but there are many genes acting together for one single phenotype. So here we can see like these clusters, like here genes are there and phenotype are there closer to each other. Here this one is above many phenotype gene embedding, so many genes here which will be governing this phenotype. So here also it goes same for both data, but what we can clearly say like phenotype embeddings are highly separated and distributed, and only genes are not the only factors which are affecting the phenotype. There can be other factors also, and this is the key observation. Marking genes on the basis of levels, and after before this we didn't mark genes on the basis of their level, whether they are active or not active, inactive, like causal or non-causal. Now we are marked genes, causal and non-causal, and phenotype embedding. We all then saw the distribution plot. So this is UMAP visualization of gene and phenotype embedding with gene levels. So we can here see like many causal genes are overlapping with phenotype embedding. So these are, but these causal genes are also overlapping with non-causal genes also. So we can say, as we talked earlier, like only not one gene is affecting the phenotype, many genes together affecting the phenotype, and maybe one certain genes which are contributing more to the phenotype. This is the case here. So in this area, at the center of the cluster, we can see at the center many genes together are acting for the phenotype, but there are certain genes which are acting more. So this is the case, and we can see that many of the phenotype embeddings are closer to other gene embeddings which are inactive, non-causal genes. So we can say that many genes which are the causal genes, they may be affecting these genes, and then these genes are causing these phenotypes. So and or we can say like there is a similarity between these phenotype embeddings and the gene embeddings. We can also say this. So this is T sign visualization of this. That was UMAP. This is T sign, and still we can see the overlapping between the causal genes and non-causal genes, and the phenotype embeddings are still distributed across the plot, and they are not getting, they are getting clustered somewhere, but not everywhere, but they are majorly they are distributed across the plot. So all the genes are acting together, not only one. And here are the conclusions and the phenotype embedding about these things. So what biological conclusion we can infer from these things, like the clear separation between causal and non-causal genes, that specific genetic factors are more closely associated with certain phenotypic traits, which could have implication in understanding the genetic influences or diseases. And there are complex interactions also, like some genes which are lying far to this phenotype, maybe these are affecting these genes, and then these genes are affecting this phenotype. This can also be the case. So we have to see, do network analysis, how one gene is affecting the whole pathway and how these phenotypes are getting expressed. Then we perform vector manipulation. For vector manipulation, we use many vector manipulation techniques, like concatenation of genes, gene phenotype embedding, then computing dot product of gene phenotype embedding, then taking difference of the vector, and in further analysis, we have taken ratio and average also. So, but before first, we try to see plot visualization of these three and analyze these three. So before this, here in this code, I have used these three concatenation, dot product, and difference of the vector, and then added the label column. Then I performed dimensionality reduction technique here, and then after reducing the dimension, I performed k-means clustering to cluster the genes, like they are active or inactive, and on the basis of their embedding, they are forming cluster, and then we visualize the genes. And after this, we saw like many things, other insights, like uh, like variance ratio and the combined cluster distribution and dot product statistics also. So here we can see the combined gene and phenotype embedding. This is our combined gene phenotype embeddings, and this is explained variance of principal component cluster distribution dot product statistics of label encoded and label encoded to both. Okay, so I'm going to explain this, and by explaining this, I will consider these factors also, explain this also. So we can clearly see in our PCA plot that the first two principal components, here we can see here that principal component these two are 7% and capturing rate of 7% and 3%, 7.9% and 3.8% around. Okay, so this is not good. So this suggests that majority of the variance is spread across many dimensions. The data is used, dimension is used, and we cannot expect that all the information is clustered in only two dimensions, in two PCAs, captured by two PCA only. So this suggests that majority of the variance is spread across many dimensions, and these two components capture only small portion of it. So showing a 2D plot, we can, seeing the 2D plot, we can also infer like much of the information is likely to be lost. So let's talk about the cluster distribution, and we talk about the cluster distribution, then we can see that PCA plot shows two distinct clusters after k-means clustering, and one cluster is more tightly packed than around the center of the plot, while the other is at more dispersed around the right side of the plot. Okay, and this was like center of the plot, and this is our right side of the plot. And label distribution, we call about like label distribution and how the labels are distributed. So we can see that plot indicates causal genes by 1 and non-causal genes by 0, and these are present in both the clusters, but are more like causal genes are present in both the clusters, we can see. But the thing is that it is more prevalent to the cluster on the right side. So causal is more prevalent in these as compared to this, and non-causal genes are dominating both sides. Like non-causal genes are higher here and higher here also. And we can see say that this distribution, non-causal genes are taking more part, causal genes taking more part here, and there are less causal genes here. Then we talk about the dot product statistics. So talking about dot product statistics, we can see the mean of the dot product. The mean of the dot product of the causal gene is 0.43186. So it is significantly higher than non-causal gene. This suggests that the average of these embeddings of the causal genes are more aligned with the associated phenotypes as compared to non-causal genes, causal genes. And if we see the standard deviation, so standard deviation of the dot product for the causal genes is 0.11, and non-causal genes is 0.06, and it is indicating that it is higher than non-causal genes. So it is indicating the variability in how causal genes align with the phenotypes. So we can see that there is a much variability in causal genes. And then if we see the distribution, then we can see like the higher quartiles, 75%, and the maximum are the causal genes. Higher quartiles are for maximum for the causal genes, like more than 50%, and also notably higher for non-causal genes also. But if we reinforce the idea that causal genes tend to have stronger alignment with phenotype. So here we after this we can see maximum and 75%. We can say that the causal genes tend to have stronger alignment with the phenotype. So if we say after seeing the statistics, we can say like 75% and maximum, like for causal genes it is high. So causal genes have stronger alignment with phenotypes. And here is the explanation of the block. And then we perform different other methods also, like vector subtraction and integrating with the causal genes. Then in this here we have took these blue and orange represent causal gene, and blue represent non-causal gene, and these are the vector embeddings, and these are the vector difference embeddings. So we can see like by taking the difference of the vectors, by taking the difference of the vectors of gene and phenotype, the data is now vector embeddings are clearly overlapping with each other. So we can say like taking differences help us to clearly separate it. Increases the similarity index because these two genes are coming, both the genes are embeddings are coming close to each other because these are overlapping. So it will be helpful by to train a model, large language kind of model, because similarity is increasing here, so it will understand how these genes are affecting these vectors, these phenotypes. Then we took vector averaging and performed dimensionality reduction. Then visualized it, so visualize the gene embedding and average vectors. Then after visualizing, we can see like many average vectors are overlapping with the genes. So average vectors and genes are overlapping. So we can say the similarity index is increasing here, and it will be helpful in training the model. So the model will take more information, and it will be able to relate the genes with the phenotypes. Then we performed element-wise multiplication and saw the distribution of PCA, UMAP, and T-sign, plot all these three, and we can see like with element-wise multiplication, PCA is not performing so well. It shows like it is very far away. So PCA might be not capturing the non-linearities well here, but UMAP and T-sign are capturing the non-linearities, global and local level features both are well. So here we can see element-wise when we multiplied took the element-wise multiplication of the gene and phenotype data, we can see the non-causal genes, causal genes are also here, and the non-causal and causal genes and the phenotype embedding. We plotted all these three, all the four. So we can clearly see phenotype embeddings are distributed across the plot, but element-wise multiplication embeddings are overlapping with our genes. So here it is doing good, but not very well because causal genes are not close with element-wise multiplication. But in T sign, we will see like the causal genes are overlapping with non-causal genes, and these causal genes are also overlapping with element-wise multiplication vectors. So here we can say that T sign is good, well representing our data distribution of how these non-causal genes and causal genes are related with element-wise multiplication vectors product we got. Then we perform PCA of the embeddings, that is multiplication wise and gene embedding, phenotype embedding. We wanted to see all three together, how the distribution of all three are together. We can see the most of the multiplication wise embedding are overlapping with our gene embeddings. So it will be if we only use gene embeddings and element wise multiplication embeddings, then our model will be able to capture well about the data, like how these two are related with each other. Then if we plot T sign, then we see like T sign is capturing very well of the whole dimensional data. When then again we see that phenotype embeddings are spread across the plot. So taking this phenotype embeddings will not be helpful for us. Here we can conclude that only taking phenotype embeddings cannot be helpful for training our large language model to predict whether which gene is causal for which phenotype and how it is affecting a phenotype. So instead of taking phenotype embeddings, we can take element-wise multiplication and gene embeddings, and then we can train model on this because these two are overlapping and sharing a larger similarity index or similar space. So the model will be able to relate more causal genes and non-causal genes with phenotypes. And here we plotted the difference of the embeddings. We can see after plotting the difference of embedding, here again PCA and UMAP are performing well. No, not UMAP and T sign are performing very well here, and the difference between the phenotype and causal genes, they are performing very well. We can say here causal genes and the differences are these overlapping with non-causal. And again, there's only one conclusion here, one steady conclusion, constant conclusion across the whole this notebook I got is only one gene is not altering the phenotype, but many genes together are expressing the same phenotype, are helping in the phenotype expression. And this represents the ratio of the genes. We took the ratio of the embeddings, so ratio of the embeddings are also performing good, but UMAP is capturing well here because UMAP causal genes and all these are overlapping, but most of the we see that most of the cluster is clustered at the center, but in T sign it is distributed across the whole graph. Then we perform clustering like k-means clustering and DB scan clustering and many clustering techniques. So after clustering, here this is k-means clustering, we perform many clustering techniques to see which genes are together and how these are affecting different phenotypes. So is there any association between the genes and phenotypes? So after clustering, we can see that k means considered as 9. We perform hyperparameter tuning here, and it considered 9 as a best parameter, 9 clusters, but these clusters are mostly overlapping. We can see. So here we can see that the cluster 4 has 1473 genes and it is affecting 28 phenotypes. So we can say it like that the cluster 4 is being affected by having 1400 genes around, and these are affecting like 28 phenotypes. And so we can say that these much genes are taking part in this much phenotypes, and only one gene can be responsible for 931. This is not possible. There can be some error. And the hierarchical clustering, for hierarchical clustering, we can say that linkage is what, and in hierarchical clustering, we can see the CDOT score is 1.5987, 0.59, 0.15987, and these are the phenotypes and genes. Like 24 genes are affecting 2414 phenotypes. So as we earlier discussed, like not only one gene is affecting one phenotype, but there are multiple genes which are affecting many phenotypes together. And in cluster 3, we can say that 20 genes are affecting 1521 phenotypes. So only saying that only one gene is causing different phenotypes is not a good, is not a better thing, but it's a cascade of genes which are acting together, affecting each other together, and then they are affecting a particular phenotype. So this is how we plotted many graph and saw, but here we can say that in DB scan, it also performed clustering, and DB scan clustering was not so good, but Gaussian mixture clustering, after Gaussian mixture clustering, we can see like 14,000 affecting 28 phenotypes. So we cannot take all these clusters are not good. Only here, only we can see hierarchical clustering is giving us best results, and it is capturing the hierarchy of gene, how all the one gene is affecting other gene, and they are affecting a particular phenotype. So in this case, we don't take all the clusters, but we consider only hierarchical clustering is good for our data representation and modeling. So that was all the analysis from my side. Thank you.